How should a cell obtain its energy? Should it perform anaerobic glycolysis, which will produce 2 net ATPs per glucose molecule, but not require oxygen and produce lactic acid as an end product? Or should it follow glycolysis with mitochondrial respiration, if oxygen is present, to produce carbon dioxide and water as an end product, producing far more ATPs? One might think that the only real variable is the presence of oxygen, so that if oxygen were not present, or if muscles were used in a type of exercise like sprinting, where oxygen might not be available fast enough, that one would rely on this anaerobic glycolysis, as opposed to if oxygen is present, or if, say, muscles are used in types of exercise where oxygen is readily available, that one would follow glycolysis with mitochondrial respiration. It turns out it's more complicated than that. The Krebs cycle, which occurs in the mitochondria, is not just about breaking down sugar for ATP. Some of the intermediates are useful in their own right. Oxaloacetate is a precursor in the synthesis of nucleotides. Citrate can be used to make fatty acids and sterols. And succinyl coenzyme A is a precursor to porphyrin, which is then the precursor to heme for hemoglobin. And so one might perform the Krebs cycle not to generate energy, but to generate the precursors for other biosynthetic pathways. In mitochondrial respiration, there are reactive forms of oxygen produced, like superoxide, anions, and hydrogen peroxide. These can also be signals which help to regulate the cell. And so, therefore, the proliferation of cells, a cell's decision to stay as a quiescent stem cell or to differentiate into a new type of cell, for example, may depend on the amount of reactive oxygen species. So, the activity of the mitochondria might not be for the purpose of generating ATP, but for rather signaling the cell, perhaps in the differentiation of the cell. A number of the intermediates in the Krebs cycle then affect methyl groups or acetyl groups being added to DNA or to the histone proteins around which DNA wraps, and thus affecting the expression of genes. And so acetyl coenzyme A, alpha ketoglutarate, succinate, fumarate, 2-hydroxyglutarate, these can all then end up altering gene expression, altering chromosome structure, unwinding DNA from histone groups, having far-reaching consequences to the cell beyond energy production. Thus, the number of mitochondria in a cell, the activity of these mitochondria, and whether or not glycolysis is followed by mitochondrial respiration is not just determined by ATP. For example, there are cells which perform aerobic glycolysis. The cells use glycolysis primarily, even though oxygen is, because that is the way in which these other aspects of mitochondrial function are best maximized for the needs of that specific cell type.